Anytime a person sits, stands, walks, or runs, muscles are contracting. Muscle cells contract and relax millions of times a day to help the body move. Let's take a closer look at what happens inside muscle tissue as muscle contraction occurs. Muscles are unique because of their specific organization system. A muscle is organized into bundles called fascicles, which are made up of muscle fibers, also called myocytes. Each myocyte contains myofibrils made up of myofilaments. Myofibrils are divided into distinct regions called sarcomeres. Each sarcomere is bordered by jagged lines called Z-lines. Within the Z-lines, two myofilaments are crucial to muscle contraction: actin and myosin. The repeated binding and unbinding of actin and myosin cause the sarcomere to shorten, otherwise known as a muscle contraction. Now that the location of muscle contractions has been identified, let's begin the process. The model scientists use to explain muscle contractions is called the sliding filament model. Before a contraction, actin and myosin are at rest in the sarcomere. At rest, two proteins, tropomyosin and troponin, block myosin from binding to actin. The nervous system controls the process of muscle contraction. Motor neurons send an action potential to the neuromuscular junction for a contraction to initiate. Neuromuscular can be broken down into two parts. Neuro refers to a neuron or nerve cell, and muscular refers to muscles or muscle tissue. This means muscle contractions begin where a motor neuron meets muscle tissue. The action potential causes the motor neuron to release a neurotransmitter, acetylcholine. Acetylcholine crosses the neuromuscular junction to initiate a muscle contraction at the myofibril. When acetylcholine enters the myofibril, sodium rushes into the muscle cell. The increase in sodium causes calcium channels to open and releases calcium into the sarcomere. Next, calcium binds to troponin, lifting itself and the tropomyosin to open actin's binding site for myosin to attach. Myosin needs energy to bind successfully to actin. In human cells, ATP provides the energy needed for cells to function. During muscle contraction, ATP molecules bind to myosin and break into byproducts of ADP and a phosphate. When ATP breaks, enough energy is released for the myosin to extend forward and grab onto the actin strand. Once myosin binds to actin, it pulls on the strand like a rope. The myosin releases the actin, ADP, and phosphate, which makes room for a new ATP molecule to attach to myosin. This process repeats over and over, causing a shortening of the sarcomere. This process is called a muscle contraction. When signaling from the motor neuron stops, the muscle has finished contracting. Calcium unbinds from the troponin and tropomyosin, causing them to block the actin strand once again. The whole process of muscle contraction may seem long, but muscle contractions occur at incredible speeds. One contraction can be a few milliseconds long, which is as fast as a camera flash or the blink of an eye. Every time muscles are used, they get stronger and can contract for longer periods of time. So whether someone is an artist, a scientist, an athlete, or a musician, muscle contractions help the body move and get stronger every day.